Good evening, everyone. So since the classes are suspended right now, I thought of recording a series of short videos so that we can make at least some amount of progress in the curriculum. So in the class we were discussing a little bit on colorimetry, on the fundamentals of colorimetry. I think before progressing any further, let us take a brief overview of what we did till now only on colorimetry. So I think we started with understanding what power distributions are, spectral power distributions are of illuminance of objects. So essentially what it means is, so for any object, for example this dark blue line, it represents the spectral power distribution of, you see, CIE illuminant A. So illuminant A is typically it simulates the light behavior, spectral behavior of your tungsten filament lamp. So it essentially spectral power distribution essentially means what amount of light, which is given in the y-axis, what amount of light is being emitted by a particular object, in this case a particular illuminant, and of what wavelength range, which is given in the x-axis. So with this basic knowledge of what spectral power distributions are, I think it's safe that we move ahead and we start reviewing the Wright and Guild experiment, which started in 1920s and helped in quantifying and measuring colors. Ultimately, what the essential criteria was, a series of standard observers, 17 standard observers together were chosen, who were asked to match a particular color that was given as a test target using 3 additive primary lights and overlapping those lights and changing the intensities of those particular lights. So once this experiment was complete, what was obtained was curves like this. One curve, what you can see quite obviously from this curve is that the red light has gone into the negative side. So this created a lot of trouble at that point in time, and I mean it's difficult to design systems having both positive and negative systems together. It makes it more computationally costly. So in order to avoid all these problems, what was essentially done was to convert the said functions or said curves, transform those said functions and curves into some functions which now at this stage only had positive values. So during the first attempt, when we had both positive and negative values, these curves were known as the CIE RGB color matching functions, whereas these particular curves where we only have positive values, these were known as the CIE XYZ color matching functions. Each curve is a color matching function, and the values that we got, each value that we got is known as the tristimulus value. So one thing is clear, that you have these curves represent what is known as the real colors, right? You can see those colors. By real I mean that these colors are derived. These X, Y and Z colors have been mathematically derived, and it doesn't exist in reality. They are only certain mathematical values. Yet what is important to note here is the fact that these XYZ values, these tristimulus values, have been created from human sensation. What I mean by this, colors are a direct, I mean these tristimulus, the human population perceives colors. In that sense, these tristimulus values, XYZ, capital X, Y, Z values are, you can say they are absolute and they're universal, because they are perceived the same way across the entire, for the entire human population. And these functions, as I told you, these functions CIE XYZ, X bar lambda, Y bar lambda, as you can see on the right hand legend, X bar lambda, Y bar lambda and Z bar lambda represents the CIE 1931 color matching functions, where all values are now positive. So now we have some background on what color spaces are, right? So this particular color space, which is a cubic 3D space, represents the RGB color space. which has 3 axes. Each axis has a maximum value of 1. That means this is the blue axis, this is the red axis, this is the green axis. So this, if I say this particular value will have, this particular point will have a coordinate of 0 1 0, this will have 1 0 0, and this will have maybe 0 0 1. So something like this. So color spaces are nothing but an imaginary coordinate system. It can be 3D, it can be 2D, where every point, every discrete point represents a unique measurable and identifiable color. So as I talked about RGB color space, so similarly the XYZ color space, which was mathematically derived from the RGB color magic functions, is also a 3-dimensional color space, so to speak. Although it is imaginary in nature, yet it is a 3-dimensional color space because it has got 3 dimensions, X, Y and Z, right? Now we can also derive, as you will be able to see in the next slide, yes, this one, you can also derive a 2D color space which is known as the YXY color space from the XY, capital XYZ tristimulus color space. This is again, I mean, I'm not going into detail of how this particular 2-dimensional color space was created after transforming the XYZ values. I mean, it has got a large mathematical explanation to it, and that is not really required at this stage for in this class to understand. So all that you need to understand is that this particular color space, which is a 2-dimensional color space, you have small x, small y. This particular color space was derived or was a derivation from the tristimulus color space, that is XYZ color space. Okay. Yet one thing is clear from this particular slide is the fact that this again is an imaginary color space, right? So I am saying these are all imaginary color space, these are all mathematical derivations, and it has got no real significant, real existence in the human visual system. Yet you can see all these colors that are present within this. So before going into these colors, let us take a look at the coordinate system that we have in front of us. So this particular coordinate system is known, what is known as the CIE chromaticity coordinate system. Why chromaticity? Obviously it represents certain chromas. So this is known as the CIE chromaticity coordinate, and this locus that you see, which is kind of of the shape of a tongue, this curve is known as the CIE spectral locus. Why spectral? You may ask the question. The reason it is known as a spectral locus is the fact that every point within this spectral locus represents a color of a particular wavelength. As you would be able to see here, it starts from 400, ends up to 700, which is your the range of the visible spectrum. So what I mean to say over here is that every point along this curve represents a unique color having a particular hue. Hue is the name of the color. So this may be red, this is orange, this is yellow, green, blue, etc. So every point along the along the spectral locus represents a color of a particular hue, right? Now what is happening if you take a look here, as you move towards the inside of this locus, you will see, so before going into this, let me answer the previous question that I already asked, is why am I saying that, although am I saying at one point that XYZ, YXY, all these color spaces are imaginary in nature, yet how can you see the colors? The answer is very simple. You see, try to think of it in this way. These are only certain values in a coordinate system, right? So for example, this point may have a coordinate value of 0.75, 0.75 and comma 0.25, right? So the color which is represented in this particular coordinate system by the coordinate values 0.75 and 0.25 is having a wavelength of, let us say, 680 or 670, like this. So similarly, any point, any coordinate point within this coordinate system will represent a particular color of a particular wavelength. So the moment you know the wavelength of that particular color, you will be able to know what, you will be able to understand what color it is. So for example, 500 to 600 roughly is represented by the color blue, and 600 to 700 is represented by the color green. 500 600 blue, 600 to 700 is green, and 600 to 700 is red in color. So just try to visualize whether what I told just now matches with what you are seeing along the points in the spectral locus. So you'll be able to get a fair bit of idea of what I am talking about. Okay, so one thing has been cleared till now, that is every point along this particular spectral locus represents a color having a unique hue. This hue is known as the pure hue of the color. Pure in the sense that this the color along the spectral locus will have the highest saturation. Why do I say highest saturation? Very simple to understand. Take any point. Let us say take this point 580, this color having a wavelength of 580 nanometers. So the moment you draw a line, or you try to draw a line joining the color which is represented by this point having a wavelength of 580 nanometers to the center of this locus. The center of this locus, center point of this locus is known as the white point. So the moment you join these 2 points by a straight line, I won't be able to draw a straight line, but you get the idea, right? Join these 2 points using a straight line. So what do you observe? You observe that as you move from the outer periphery, that is the spectral locus itself, and you move towards the white point of this locus, the saturation of the particular color decreases. So here let us say this color is orange. So the moment you go inside and the moment you reach the white point of this locus, you will see this particular point is having no saturation at all. That is why this is known as the region of this is known as the white point, and this particular point is having the highest saturation. That is why this point is known as the point of pure hue of this particular color, right? So okay, so just to recap what we learnt till this point is the fact that this has been derived from the CIE Tristan Lewis value. This is a 2D representation of that color space, where the outer periphery of this particular locus denotes colors of pure hues. As we move towards the towards the white point, it represents color of reduced saturation. So this particular color space gives us an idea about the hue of the color. So my question to you would be to tell me, how can I refine this particular model in order to include the lightness data into it? So that would be maybe something for you guys to find the answer to. So the disadvantage of this particular color space is the fact that although we have hue and saturation, yet we do not have the lightness value. As I told you in the class already, that X and Z, that is capital X and capital Z together, they denote the hue of the particular color, and capital Y denotes the lightness. Since the lightness value is not present in this particular color space, that is why this color space is also known as the YXY. So Y represents the lightness value which is not being represented by this color space. Okay, so yeah, so this was about the CIE YXY color space and spectral locus. So this is you can say this is a tool using which we will be able to measure colors. How will I be able to measure colors? Now if I have certain formulas with me or some guide with me using which I will be able to find out the chromaticity coordinate of that particular color. So for example, if I ask you, using this curve, tell me what will be the approximate color of a or what will be the approximate nature of a color which is represented by the chromaticity coordinate 0.5, 0.3. So this is my question to you. What will be the nature of the color? Ultimately, what is the hue? I am asking what I am essentially asking to you is what will be the hue of the color? Whether it will be red, green, blue, orange, yellow, whatever. What will be the hue of the color where whose chromaticity coordinate is 0.5, 0.3. Again I am repeating the question. What will be the hue of the color having chromaticity coordinate 0.5, 0.3? So this is if you can answer this question, you have essentially figured how to read this particular chart, right? So this, as you can understand, that this is nothing but a tool using which we will be able to quantify and measure a particular color. So if the designer tells me that, see, my logo uses the color having chromaticity coordinate 0.5, 0.3, 0.5 and 0.3. So immediately, whether you will be able to, you are using a Mac computer or you are using a Windows based computer, doesn't matter. Automatically, this is universal system. Automatically, once you have the chromaticity coordinate of a particular color, you will be able to reproduce the color. Now, naturally, you will ask the question, how do I reproduce the color? I agree, I have that chromaticity coordinate of a particular color, but how do I reproduce that color? In order to understand or answer this question, we need some other information also that we will be discussing in the later slides. But you get the basic idea, right? That once we have the chromaticity coordinate of any color within the spectral locus, I will be able to reproduce the color. This is the first point that you should note. The second point you should also try to understand or try to realize by now is the fact that this is also, as I told you in the class last day, that there are essentially two types of color spaces. One is device dependent and another is device independent. So what do you think will be the nature of this color space? It is obviously a device independent color space. Why? If I ask you, why is this YXY spectral locus or color space device independent? Because where has it been derived from? কোথা থেকে এসেছে এই color space? It has come from the CRE capital XYZ, that is CRE tristimulus color space, right? And we also know at this stage that the CRE XYZ color space is a device independent color space because it is derived from how human population perceives color. It is not dependent on a particular device. It is not dependent on whether I have a Mac computer or a Windows computer or I am printing using a HP printer or a Heidelberg 4-color offset printer or a Epson inkjet printer. It doesn't matter. It is XYZ values, or in this case, as I was talking about on YXY values, these values are absolute values, absolute colorimetric values. Why absolute? In what sense is it absolute? Because they do not change from person to person. They do not change its value when because why don't they change? They do not change because they have been derived from how humans perceive color, how human cones perceive particular color, which is universal across the human population. So since this is the first video, so I would like to end. I don't know whether you will be able to see or watch this video and able to understand anything from the video. I don't know. So my suggestion to you is, you may send me a mail. If you watch this video, send me a mail. This is the first time I am doing this, okay? So send me a mail writing me to maybe incorporate any other changes while doing these video lectures. So just send me a mail or give me your